patients, biochemicals that indicate that inflammation is occurring, inflammatory markers, are elevated and can even be used to predict cognitive decline and the development of dementia. Among the most famous of these biochemicals are the cytokines, small proteins released by cells that affect the behavior of other cells and are often important participants in the inflammatory process. C-reactive protein, interleukin 6, and tumor necrosis factor alpha are all cytokines. We now have the ability to image the brain in ways that allow us to see these inflammatory chemicals in action, so we can identify direct correlations between the degree of inflammation and the degree of cognitive impairment. Tumor necrosis factor alpha in particular seems to play an important role in inflammation throughout the body, and in addition to being elevated in the blood of Alzheimer's patients, it's been found to be elevated in a variety of other inflammatory conditions including psoriasis, rheumatoid arthritis, cardiovascular disease, Crohn's disease, and asthma. So important is the role of tumor necrosis factor alpha in these conditions that pharmaceutical companies are investing huge sums of money to try to develop ways of reducing it. The global market for tumor necrosis factor inhibitors today exceeds $20 billion annually. In some people, specific genes can naturally increase inflammation, and they can further raise one's risk for developing diseases rooted in inflammation. But genetic factors are far from the whole story. You can do a lot to influence the expression of your genes, from turning off or suppressing bad genes to flipping the switch on good genes that will help assure your health. In Grain Brain I explored in depth one of the most influential and fundamental ways one can support the expression of those good genes while suppressing the bad and, in turn, keep inflammation in check when it's not needed for survival, by maintaining healthful blood sugar levels. Elevated blood sugar stirs up inflammation in the bloodstream, as excess sugar can be toxic if it's not swept up and used by cells. It also triggers a reaction called glycation, the biological process by which sugar binds to proteins and certain fats, resulting in deformed molecules that don't function well. These sugar proteins are technically called advanced glycation end products. The body does not recognize ages as normal, so they set off inflammatory reactions. In the brain, sugar molecules and brain proteins combine to produce lethal new structures that contribute to the degeneration of the brain and its functioning. The relationship between poor blood sugar control and Alzheimer's disease in particular is so strong that researchers are now calling Alzheimer's disease type 3 diabetes. Although the studies documenting this phenomenon go back about 10 years now, neuroscience is further crystallizing the big picture. We are finding out that alterations in the gut microbiota pave the way for the development of diabetes and proliferation of ages and therefore increase the risk for Alzheimer's disease. I'll detail how this is possible in Chapter 4, but here's a quick primer. In 2012, the journal Nature published a study showing that people with type 2 diabetes suffered from bacterial imbalances dysbiosis in their gut. These imbalances cause them to lack important byproducts from the gut bacteria that are needed to maintain the health of cells in the digestive system. Bear in mind that people who have type 2 diabetes are under a lot of metabolic stress because their bodies cannot transport glucose successfully from the bloodstream into their cells. And in areas of the body that don't have a glucose transport system, such as the nerves and brain, scientists can identify other forms of metabolic stress such as those ages, which can result in conditions including peripheral neuropathy, weakness, numbness, and pain caused by damaged nerves, and damage to blood vessels and brain function. This discovery has been groundbreaking in my world. To know that at the center of the cascade of events leading to diabetes and brain disease is a disrupted gut community is, at least to me, staggering. I love how a group of Chinese researchers recently explained the facts of the matter in a report published in the esteemed Food Science and Human Wellness Journal. Great progress has been made in the field of the resident microbiota in type 2 diabetes in recent years. Microbiota contribute not only to low-level inflammation in the onset of type 2 diabetes, but also to the further development of type 2 diabetes through inflammatory components. It has also been extended to various type 2 diabetes-related complications, including diabetic retinopathy, kidney toxicity, atherosclerosis, hypertension, diabetic foot ulcers, cystic fibrosis and Alzheimer's disease. These studies together support the crucial role of microbiota in maintaining the intestinal barrier integrity, sustaining a normal metabolic homeostasis, protecting the host from infection by pathogens, enhancing host defense systems and even influencing the nervous system in type 2 diabetes. The researchers went on to discuss the influential role of dietary choices in changing the microbiota for the better and reducing risk for these conditions. They also pointed out that various herbs and supplements with known anti-diabetic properties work to control blood sugar through the microbiome. In other words, they may not necessarily be directly affecting insulin and glucose, rather, they are positively affecting the microbiome. For example, the traditional Chinese herbal components berberine and ginseng, as well as the compounds found in tea, coffee, wine, and chocolate have anti-diabetic qualities through their effects on gut bacteria. These compounds either change the composition of gut bacteria for the better or are metabolized by the gut bacteria before being absorbed into the body. After thousands of years, ancient Chinese herbal practices are finally getting the explanation they deserve. It's the gut bugs that are making use of these herbal compounds first so we can benefit from them. Dr. James M. Hill is a senior scientific investigator and professor of neuroscience at Louisiana State University School of Medicine. His laboratory is among many high-tech labs connecting the dots between the gut's microbiome and the risk for brain disease. He recently published a report outlining the multiple ways in which the brain and its functioning are influenced by what goes on in the gut. In his studies using mouse models, he's explored how good gut bacteria are capable of producing important brain chemicals like BDNF, gamma-aminobutyric acid GABA, and glutamate. And levels of these important chemicals directly reflect what goes on in terms of gut bacteria. When researchers disrupt gut bacteria in mice, they not only observe behavioral changes in the mice, but also calculate changes in the volume of these chemicals. Previously, I described brain-derived neurotrophic factor as a critical brain growth protein. BDNF is involved in neurogenesis, the process by which new neurons are created. BDNF also protects existing neurons, ensuring their survival and encouraging connections, or synapses, between them. This synapse formation is essential for thinking, learning, and higher levels of brain function. Decreased levels of BDNF are found in a panoply of neurological conditions, including Alzheimer's, epilepsy, anorexia nervosa, depression, schizophrenia, and obsessive-compulsive disorder. And while we know that BDNF can be increased through aerobic exercise as well as by consuming the omega-3 fat DHA, we are now learning that this critically important brain chemical is vitally dependent on the balance of bacteria that live within the gut. In the American Medical Association's journal Neurology, a fascinating report by a team at Boston University School of Medicine, published in November 2013, revealed how blood levels of BDNF relate to risk for developing dementia. The study culled information from the now famous Framingham Heart Study, one of the largest epidemiological studies ever performed, to look at blood levels of BDNF in a group of 2,131 adults. These individuals were free of dementia at the start and were followed for up to 10 years. What these Boston University researchers found was that those individuals at the beginning of the study who had the highest levels of BDNF had less than half the risk of dementia compared to those with the lowest BDNF levels. They stated that BDNF may also be reduced in healthy people who are destined to develop dementia or Alzheimer's disease. The researchers concluded the obvious: our findings suggest a role for BDNF in the biology and possibly in the prevention of dementia and Alzheimer's disease. GABA, another important chemical manufactured by the gut bacteria, is an amino acid that serves as a neurotransmitter in the central nervous system. It's the main chemical messenger in your brain that calms down nerve activity by inhibiting transmissions and normalizing brain waves. In other words, GABA returns the nervous system to a more stable state so you can weather stress better. In 2012, researchers at Baylor College of Medicine and Texas Children's Hospital identified a strain of bifidobacteria that secretes large amounts of GABA, suggesting that it may play a role in preventing or treating not just brain disorders but inflammatory bowel disorders such as Crohn's disease. Because GABA mutes neuronal activity, it keeps in check anxiety, of course, being a common trigger to gastrointestinal disorders rooted in inflammation. Glutamate, yet another vital neurotransmitter that is produced by the gut bacteria, is involved in most aspects of normal brain function, including cognition, learning, and memory. It's abundant in a healthy brain. A slew of neurological challenges, from anxiety and behavioral deficits to depression and Alzheimer's disease, have been attributed to a lack of GABA and glutamate. One of the most important takeaways from the new research on the connection between microbes and brain health is that disruption isn't just about having a misbalanced microbiome, in which the thugs outnumber and trample on the good guys, triggering inflammation and robbing the body of vital materials produced by those good guys. In millions of people today, the gut is largely disrupted by increased intestinal permeability, which fuels a continuous state of low-grade inflammation. Let me break this down for you. Now discussion on topic, titled, The Perils of a Leaky Gut. Your gastrointestinal tract, from the esophagus to the anus, is lined with one single layer of epithelial cells. This cellular layer represents a critical interface between you and your environment inside versus outside. In fact, all of the body's mucosal surfaces, including those of the eyes, nose, throat, and gastrointestinal tract, are a large point of entry for various pathogens, so they must be well protected by the body. These surfaces are lined by a mucous membrane, a type of tissue that secretes mucus, hence the term mucosal. The intestinal lining, the largest mucosal surface, has three main functions. First, it serves as the vehicle or mechanism by which you obtain nutrients from the foods you eat. Second, it blocks the entrance into the bloodstream of potentially harmful particles, chemicals, bacteria, and other organisms that can pose a threat to your health. Third, it contains chemicals called immunoglobulins that bind to bacteria and foreign proteins to prevent them from attaching to your gut's lining. These chemicals are antibodies that are secreted from immune system cells on the other side of the gut lining and that are transported into the gut via the intestinal wall. This ultimately allows pathogenic organisms and proteins to move on and be excreted. There are two pathways that the body uses to absorb nutrients from the gut. Using the transcellular pathway, nutrients move through the epithelial cells, and using the paracellular pathway, nutrients pass between the epithelial cells. The connection between cells, called a tight junction, is an intricate and highly regulated affair. When we are talking about permeability issues in the gut, or the so-called leaky gut, we are referring to problems in the competency of these tight junctions, which measure between 10 to 15 A ring. A ring is the abbreviation for angstrom, a unit so small that the only way to picture it is to think of a microscopic space millions of times smaller than the head of a pin. It's much smaller than a typical virus or bacterium. If the junctions aren't working properly, they fail to appropriately police what should be allowed to pass nutrients or kept out potential threats. As the gatekeepers, these junctions determine, to a large extent, the set point of inflammation, your body's baseline level of inflammation at any given time. It's well documented now that when your intestinal barrier is compromised, you are susceptible, through increased inflammation, to a spectrum of health challenges, including rheumatoid arthritis, food allergies, asthma, eczema, celiac disease, inflammatory bowel disease, HIV, cystic fibrosis, diabetes, autism, Alzheimer's, and Parkinson's. Theory, by Cyrex Labs, measures antibodies produced by the immune system when it is confronted by a molecule called LPs, short for a lipopolysaccharide. No conversation about the microbiome, inflammation, and brain health can exclude the impact of this molecule. Now discussion on topic, titled, LPs, the incendiary device. If there were ever a clear biological villain that flips on inflammatory pathways in the body, it would be lipopolysaccharide. It is a combination of lipid fat and sugars and is a major component of the outer membrane of certain bacteria. In addition to providing a lot of the structural integrity of the bacteria, LPs also protects these bacteria from being digested by bile salts from the gallbladder. LPs protected bacteria, which are called gram-negative bacteria, that is, a class of bacteria that do not retain the purple stain used in the gram staining method used to differentiate bacteria, are normally abundant in the gut, representing as much as 50 to 70% of the intestinal flora. We've long known that LPs, which is classed as an endotoxin, meaning a toxin that comes from within the bacteria, induces a violent inflammatory response in animals if it finds its way into the bloodstream. LPs is used experimentally in research to instantly create inflammation in laboratory studies. Animal models that are used for such diverse conditions as Alzheimer's disease, multiple sclerosis, inflammatory bowel disorders, diabetes, Parkinson's disease, amyotrophic lateral sclerosis (ALS), rheumatoid arthritis, lupus, depression, and even autism employ LPs because of its ability to quickly push that inflammatory button in the body. This ultimately allows researchers to study these ailments and examine their relationship to inflammation. Human studies show that elevated markers of LPs are seen in many of these conditions. Normally, LPs is blocked from the bloodstream by the tight junctions that exist between the cells lining the intestine. But, as you can imagine, when those junctions are compromised and the lining becomes leaky or permeable, LPs goes into circulation and can fuel inflammation and inflict damage. So LPs levels in the blood are indicative not only of inflammation in general but also of leakiness of the gut. In one of the most alarming studies ever done on LPs, grad student Marielle Suzanne Kahn and her colleagues at Texas Christian University showed that injections of LPs into laboratory animals' bodies, not brains, led to overwhelming learning deficits. In addition, these animals developed elevated levels of beta amyloid in their hippocampus, the brain's memory center. Beta amyloid is a protein that is strongly implicated in Alzheimer's disease pathology. Researchers are now investigating ways that beta amyloid can be reduced in the brain or even prevented from forming. Bottom line, elevated LPs in the blood could be a big contributor to the increased beta amyloid in the brain that's so characteristic of Alzheimer's disease. Other studies have demonstrated that mice who receive injections of LPs in their abdomens experience severe memory problems. LPs has also been shown to decrease production of BDNF. Moreover, we now have evidence that there's 3 times as much LPs in the plasma of Alzheimer's patients as in healthy controls. This is powerful information that once again speaks to the gut-brain connection and the impact of inflammation and gut permeability. It's a given that we all have LPs in our guts because it's an important structural component in a lot of the bacteria there, but it shouldn't be a given that it ends up in the bloodstream, where it can be quite destructive. ALS, also known as Lou Gehrig's disease, is a devastating, almost universally fatal condition for which there is no cure and just one only modestly effective FDA-approved drug. It affects more than 30,000 Americans. Research is now looking at the role of LPs and gut permeability in this disease. Not only are there higher levels of LPs in the plasma of ALS patients, but the level of LPs directly correlates to the severity of the illness. This new data has led some experts to wonder if a chief instigator of ALS isn't in the brain or spinal cord, but in the gut. In other words, researchers may have been looking in the wrong place all these years. The evidence against LPs and the inflammation it causes is so incriminating that scientists at the University of San Francisco have stated that this knowledge may represent novel targets for therapeutic intervention in patients with ALS. One more example showing the power of LPs, Dr. Christopher Forsyth and his colleagues at Rush University Medical Center in Chicago have explored LPs and intestinal permeability with regard to Parkinson's disease, and they have indeed found direct correlations. Patients with Parkinson's disease show much higher levels of LPs than healthy controls. And in the next chapter, we'll see how the latest science on depression shows that elevated LPs is among the most villainous suspects in triggering the mood disorder. Now discussion on topic, titled, Brain Health Begins in the Gut. So, you should be able to draw the same conclusion I'm going to make. We really have to pay attention to how our gutsy tribes are fed and nurtured. We also have to ensure the integrity of the gut lining. In my previous book, I outlined the primary instigators of inflammation in the body that can interfere with neurological function and brain health, ubiquitous ingredients like gluten and sugar, and a lack of antidotes such as healthy fats, exercise, and restful sleep. But now we have more science to show that the story doesn't just start with an inflammatory response to bread and linses. It starts with an upset microbiome and the disastrous effects of molecules like LPs that go rogue once in circulation. As you'll be learning more about in upcoming chapters, factors such as antibiotics and other medications, chlorinated water, certain foods, and even stress all play a part in determining the diversity and balance of the gut bacteria and, therefore, the set point of inflammation. Not only do gut microbes influence the environment in your body, but they contribute to that environment by producing certain chemicals that affect the health of the brain and entire nervous system. They determine the strength and fortitude of your gut wall. And they can even produce various vitamins that are essential to brain health, including B12. It's well substantiated that low levels of B12 are a huge risk factor for dementia, not to mention other neurological challenges such as depression. I can't tell you how many times I've seen remarkable improvements in a patient's clinical depression just through B12 supplementation. Research shows that B12 deficiency here in America affects 10 to 15% of people over the age of 63 and may well have to do with changes in the gut bacteria as a consequence of poor diet and the medications they take in an attempt to remain healthy. The connection is straightforward. B12 synthesis in the body occurs primarily in the small intestine, where gut bacteria manufacture it using cobalt and other nutrients. Although vitamin B12 can be obtained from the diet, mostly from animal foods including fish, meat, poultry, and eggs, some of the B12 that gets absorbed in the gut to meet your daily requirements comes from those bacterial factories. I can't reiterate this enough, the health and variety of your belly's bugs directly depend on the foods that you eat. Foods that are high in fiber, which provide fuel to the gut bacteria, and reduced in refined sugars support a robust melange of bacterial species, which helps maintain the integrity of the gut wall, keep blood sugar in check, reduce inflammation, and manufacture all those important substances and molecules critical for brain health and function. Moreover, there's a big difference between fats that fuel inflammation and fats that help control inflammation. Omega-6 fats dominate in the Western diet today. These are the pro-inflammatory fats found in many vegetable oils that have been linked to an increased risk for brain disorders as well as heart trouble. Omega-3 fats, on the other hand, such as the fats found in olive oil, fish, flaxseed, and wild grass-fed animals, boost brain function, help stamp out inflammation, and can actually counterbalance the detrimental effects of the omega-6 fats. Anthropological research reveals that our hunter-gatherer ancestors consumed omega-6 and omega-3 fats in a ratio of roughly 1:1. Today we consume an astronomical 10 to 25 times more omega-6 fats than those ancestors did. Let's briefly turn to the notion of coffee as protective of the brain, as this will further convince you of the power of your dietary choices in light of your gut bacteria. A recent publication in the Journal of Alzheimer's Disease revealed an impressive risk reduction for the development of the disease in people who drink coffee. The study, carried out in Finland in collaboration with the Karolinska Institute, followed 1,409 individuals aged 65 to 79 years for an average of 21 years. People who drank between 0 and 2 cups daily were characterized as being low coffee drinkers. Those who drank between 3 and 5 cups were deemed moderate, and those who downed more than 5 cups a day were placed in the high category. Moderate drinkers at midlife showed an incredible 65% decreased risk for developing Alzheimer's in comparison to low drinkers. Although the people who drank more than 5 cups a day also were at reduced risk of dementia, not enough people comprised this group to draw statistically significant conclusions. Lead researcher Dr. Mia Kivipaldo, professor of clinical geriatric epidemiology at Karolinska, commented on her study by stating, "Given the large amount of coffee consumption globally, the results might have important implications for the prevention of or delay the onset of dementia/Alzheimer's disease. The findings need to be confirmed by other studies, but it opens the possibility that dietary interventions could modify the risk of dementia/Alzheimer's disease." I'd take this a step further. Researchers are just beginning to unravel the brain protective attributes of coffee, and the latest research plainly demonstrates that it happens at the level of the microbiome. The laboratory research is now extensive, in fact, and unambiguously shows that, through the workings of the belly's bugs, coffee reduces the risks of type 2 diabetes, strokes, Alzheimer's disease, Parkinson's disease, and even cancer and cardiovascular disease. It does this through an array of mechanisms that include the gut bacteria. For starters, intestinal bacteria can easily digest the coffee bean fiber that remains in the brewed liquid, extracting its energy for their own growth and health. It also has been shown to reduce the ratio of Firmicutes bacteria to Bacteroidetes bacteria, and later on we'll see how this shift in ratio is associated with reduced risk for diabetes and obesity and, therefore, reduced inflammation as well. What's more, we also understand that coffee is a rich source of polyphenols, molecules with known healthful properties. They are the most abundant antioxidants in the human diet. It has been estimated that we consume as much as 1 gram of polyphenols each day, which is about 10 times higher than daily consumption of vitamin C and 100 times higher than our daily intake of vitamins E and A. Polyphenols are not only found in coffee. They permeate red wine as well as other foods and have become a major focus of research. But here's the crux of the matter: the ability of your body to extract and use the polyphenols you consume is dictated largely by the gut bacteria. Those bugs once again take center stage in coordinating your biology for the benefit of your health. In order to fully gain the health benefits of polyphenols from the foods you eat, you need a healthy microbiome. The top 3 ways your gutsy friends reduce your risk of brain disease. First, they help control inflammation. The balance and diversity of gut bacteria regulates how much inflammation occurs in the body. Healthy levels of a variety of good bacteria limit the production of inflammatory chemicals in the body and in the brain. Inflammation, as you already know, is the basis for degenerative conditions in the human body, including diabetes, cancer, coronary heart disease, and Alzheimer's disease. Second, they bolster the intestinal wall's integrity and prevent gut permeability. A leaky gut brought on by an imbalance of gut bacteria allows various proteins normally found in the gut to make their way through the gut wall and challenge the immune system. This scenario turns on an immune response that again leads to inflammation. We now recognize that a variety of factors increase gut permeability, including certain medications, pathogenic bacteria, stress, environmental toxins, elevated blood sugar, and ingredients like gluten. Third, they produce important chemicals for brain health, including BDNF, various vitamins such as B12, and even neurotransmitters like glutamate and GABA. They also ferment certain foodborne compounds like polyphenols into smaller anti-inflammatories so they can be absorbed into the bloodstream and ultimately protect the brain. Now discussion on topic, titled, Inflammation, the Gut, and the Mighty Mitochondria. To bring the conversation about inflammation full circle, we must take a closer look at how our mitochondria serve us. These are the tiny organelles, found in all cells except red blood cells, that generate chemical energy in the form of ATP adenosine triphosphate. They have their own DNA, and the current thinking is that they originated from bacteria. They were once free-living bacteria that ultimately took up residence within our cells and provided our cells the benefit of producing energy. Like bacterial DNA, the DNA of the mitochondria is arranged in a circle, quite unlike the genetic material found within the nucleus of the cell. We now recognize that these intracellular organelles do far more than simply make energy. Mitochondria exert significant control over nuclear DNA. Given their bacterial origin and their unique DNA, mitochondria should be considered part of the human microbiome. Healthy mitochondria make for a healthy human. And we now know, for instance, that they play a key role in degenerative diseases such as Alzheimer's, Parkinson's, and even cancer. First observed by the German Dr. Carl Binde in 1897, these intracellular particles appeared as tiny thread-like grains. Hence the name mitochondria, derived from the Greek mitos, meaning thread, and chondron, meaning grain. As an aside, while the nucleus of the cell contains exactly 2 copies of its DNA, the mitochondria may have anywhere from 5 to 10 copies of its DNA. But it wasn't until 1949 that the role of mitochondria as cellular powerhouse was fully explained by 2 American scientists, Eugene Kennedy and Albert Leininger. Basically, these organelles are able to use carbohydrate fuel and convert it to energy that powers most cell functions. The energy produced by this reaction is called oxidative metabolism because oxygen is used up in the process, as it is in a fire. But mitochondrial respiration differs from a fire in that rather than releasing energy in an uncontrolled reaction, mitochondrial energy is stored in a unique molecular form called ATP. Energy-rich ATP can then be delivered throughout the cell, releasing energy on demand in the presence of specific enzymes. Individual cells of the brain, skeletal muscle, heart, kidney, and liver may contain thousands of mitochondria each, to the extent that in some cells, up to 40% of the material is made up of mitochondria. According to Professor Enzo Nicoli of the University of Milan, you and I each possess more than 10 million billion mitochondria, making up a full 10% of our body weight. A fundamental fact to understand here is that the use of oxygen in the process of producing energy provides a high level of efficiency. While cells do have the ability to utilize other chemical pathways to produce ATP in the absence of oxygen, this process, anaerobic metabolism, is only 1/18 as efficient as oxidative metabolism. But with the use of oxygen comes a price. An important byproduct of the work done by mitochondria is the production of chemicals, related to oxygen, called reactive oxygen species RAS. RAS are more commonly known as free radicals. In a strict scientific sense, the term free radicals refers not only to reactive oxygen species, but also a similarly reactive family of radicals, reactive nitrogen species. For purposes of simplification, and as has become the norm in non-scientific publications, we use the term free radicals to refer to reactive oxygen species. Most everyone is familiar with the term free radicals today, for they've been widely described in lay literature, from beauty magazines to advertisements for anti-aging skincare products. Although they are often demonized for the negative effects they can have in the body, free radicals have a positive role in human physiology in a number of ways. They take part in regulating apoptosis, the process by which cells undergo self-destruction suicide. While it may seem puzzling at first as to why cellular suicide should be looked upon as a favorable event, apoptosis is a critical and necessary cellular function. The term apoptosis was first ascribed to Hippocrates and initially taken to mean the falling off of the leaves. But it was not until a publication in the British Journal of Cancer in 1972 by Alistair R. Curry that the term gained traction in the scientific community. Thereafter, researchers used the word to describe the process by which cells are intentionally eliminated. Without apoptosis, for example, we wouldn't have individual fingers, for it's this very process by which our digits can take shape from the limb buds during our embryological development, thus allowing our hands to form from what initially appear as mittens. Apoptosis is critically important, as it allows our body to rid itself of the multitude of cancer cells that appear spontaneously within us. Each day 10 billion cells are terminated to make way for newer, healthier cells. And free radicals created by mitochondria during the process of energy production play a key role in this process. As with so many things in life, apoptosis does have its dark side. While there are many situations in which activating the demolishing genes of a cell is a good thing, when mitochondrial function becomes impaired, cellular suicide can be induced in otherwise normal, healthy cells. In fact, this is a fundamental mechanism leading to the destruction of neurons and neurodegenerative conditions such as Alzheimer's disease, multiple sclerosis, Parkinson's disease, and Lou Gehrig's disease. Brain cell apoptosis isn't just limited to these disease processes, though. It's occurring in each of us throughout our lifetimes and is responsible for our general decline in brain function as we age. Until quite recently, scientists subscribed to the paradigm that all cellular functions, including apoptosis, were directed by the cell's nucleus. But as British biochemist Nick Lane notes in his compelling book, Power, Sex, and Suicide, there has been a change in the emphasis that amounts to a revolution, overturning the nascent paradigm. The paradigm was that the nucleus was the operations center of the cell, and controls its fate. In many respects this is of course true, but in the case of apoptosis it is not. Remarkably, cells lacking a nucleus can still commit apoptosis. The radical discovery was that mitochondria control the fate of the cell, they determine whether a cell shall live or die. Mitochondria, then, are much more than simple organelles that turn fuel into energy. They wield the sword of Damocles. And it should come as no surprise that they can be damaged very easily by inflammation, especially the kind that originates from disorderly conduct in the gut's microbial community. The gut, after all, is an origin of inflammation given the complex interplay between its microbial inhabitants and the immune system. So inflammatory processes regulated by gut bacteria and resulting in inflammatory molecules coursing through the bloodstream, reaching cells and tissues, will assault the mitochondria. Moreover, byproducts of an imbalanced gut community can also inflict direct damage to mitochondria and in turn fuel more inflammation. Studies are currently underway to investigate the link between the human microbiome and mitochondrial diseases, especially those that can be passed on to future generations. Mitochondrial diseases include a group of neurological, muscular, and metabolic disorders caused by dysfunctional mitochondria. Disorders as diverse as diabetes, autism, and Alzheimer's have all been linked to mitochondrial problems. In Chapter 5, we'll see how mitochondrial dysfunction in children with autism is providing clues to this condition, as well as to the role that the gut bacteria might have in the development of the brain disorder. With this understanding of the value of mitochondria, it's exciting to know that new mitochondria grow all the time. Perhaps more importantly, we can make lifestyle changes to increase the growth of mitochondria, a process called mitochondrial biogenesis, and thereby enhance an important part of the human microbiome. Lifestyle factors that stimulate this process include eating a diet that derives more energy or calories from fat than from carbohydrate, a central theme of Grain Brain, reducing your caloric intake, and engaging in aerobic exercise. Later on, in the book, I'll go into greater detail about the things you can do to enhance your mitochondria and, in turn, your entire microbiome. There is one more attribute of the mitochondrial DNA that, to me, has profound implications. All of the mitochondrial DNA is inherited solely from the female lineage. During reproduction, while the nuclear DNA of the sperm joins with that of the egg, the male's mitochondria are excluded. Think of it. Mitochondria, the source of energy to sustain our lives, are the embodiment of a purely feminine genetic code. This concept has prompted scientists to conceive of a mitochondrial Eve, the first human mother from whom all humans have derived some of their mitochondrial DNA. Mitochondrial Eve is thought to have lived some 170,000 years ago in East Africa at a time when Homo sapiens was developing as a species separate from other hominids. When you consider the fact that bacteria were the planet's original inhabitants, it's no surprise that by the time humans came along, multicellular organisms had long since forged a symbiotic relationship with many bacteria, some of which eventually landed within our own cells to establish a third dimension to our genetic fingerprint. Now discussion on topic, titled, Gaining Control of Mysterious Illnesses. Perhaps one of the most extraordinary examples of harnessing the power of the gut bacteria to address inflammatory diseases that impact the brain is seen in a patient such as Carlos. Carlos, 43, came to see me in June 2014. He needed a cane to stand and had episodes of feeling as if his legs wouldn't work and as if he could lose his balance easily. When I asked him about his medical history, he told me about one morning back in 1998 when he woke up feeling drunk and dizzy. When he went to see a neurologist, an MRI scan of his brain was performed, but the results came back normal. Carlos remained unsteady for the next 2 weeks and then began to feel better. 2 weeks after that, while exercising, he felt as if ants were crawling down his back. His vision blurred and, hoping to find another opinion about his symptoms, he went to see a naturopath. That's when he began taking various nutritional supplements, and he felt a little better thereafter. 3 years later, he had the sudden onset of numbness in both legs from the waist down. Again, he was given a new round of nutritional supplements, and after 3 more months, he felt somewhat improved. 2 years later, he had another episode, and this similarly resolved itself with more supplements. In 2010, however, he began noticing a progressive decline in his balance and, despite various nutritional supplements, his deterioration continued, rapidly. By 2014, Carlos went through more tests with a neurologist, including another MRI scan of his brain. And this time, his results revealed aggressive abnormalities, especially in the deep white matter of his brain in both hemispheres and even in the brainstem. These findings, in addition to abnormalities noted in an MRI of his cervical spine, a lumbar puncture, and electrical testing results, all pointed to a diagnosis of multiple sclerosis. MS is an inflammatory disease characterized by damaged nerves in the brain and spinal cord. The insulating covering of these nerve cells, called the myelin, becomes impaired, and so the nervous system breaks down, leading to a wide range of symptoms, physical, cognitive, and even psychiatric. Scientists have long puzzled over what causes multiple sclerosis, though it's generally thought to result from a glitch in the immune system. We just don't know what triggers this glitch, which leads the body to attack its own nerve cells. And yet epidemiological studies have determined that living in an urban environment is a significant risk factor for having this autoimmune disease, similar to the increased risk for developing Alzheimer's disease in urban, westernized settings. Could multiple sclerosis, and many other neurological conditions, be directly related to changes that have occurred in the gut's bacterial community? Over the past several years, I've noticed that MS patients almost always were either born by C-section, not breastfed, or treated with antibiotics for some illness early in life. In fact, new research published in 2013 showed that the risk of MS was reduced by 42% in people who were breastfed. In reviewing Carlos's early life experience, once again I found the same pattern to his history. He'd only been breastfed for several days. I explained to Carlos that we now understand more about the role gut bacteria play in the immune system, and that recent animal research clearly had identified changes in gut bacteria as possibly playing an important role in this disease.